All right, well, hello everyone, and thanks to the organizers for having me speak today, and thanks to you for showing up on a Sunday morning to listen. I'm Stacey Kigar, a postdoc here in the departments of medicine and psychiatry, and I work under the supervision of Ed Bullmore. You can probably tell from my accent that I'm an American. I've actually only been in Cambridge for about 6 months. So for the next hour or so, I'll be talking to you mostly about work done during a previous fellowship at the National Institutes of Health in the Washington, D.C. area. And I've included what I find to be a pretty spectacular photo of the cherry blossoms. And someday when we're in a post-COVID world, I hope that you get to check out the Cherry Blossom Festival because it's really pretty beautiful. Diving right in, the title of my talk is perhaps a bit technical, but the question I'm really interested in is what mechanisms cause psychiatric disorders like depression, and how can we fix the problem? I'm sure it comes as no surprise to any of you that major depressive disorder, or MDD, is a major public health issue. It affects more than 250 million people globally, according to the World Health Organization. What you may not know is that as many as 30% of patients treated for depression do not respond to antidepressant drugs. That's 75 million people who aren't being effectively helped. The pharmacological tools that physicians currently have are all basically working in the same way. That is, by increasing the availability of serotonin in the brain. But for the 75 million people with treatment-resistant depression, serotonin deficiency can't be the only explanation for their symptoms. So it is abundantly clear that we need new strategies to combat MDD. One interesting theory posits that there is a relationship between chronic inflammation and depression. And by the way, for further reading, I'd highly recommend this book by my boss. I've listed a few clinical studies here that support this idea that there is an inflamed mind hypothesis, including the existence of comorbidities between autoimmune disorders and mood disorders. But going one step further, in this study, the authors found a relationship between increased peripheral inflammation and treatment-resistant depression, which is really interesting. Further, recent work from Ed's group broadly characterized circulating white blood cell types from depressed patients and compared them to healthy controls. And in this study, they uncovered what they called an inflamed subgroup of depressed patients. They also found a relationship between certain classes of white blood cells, particularly neutrophils, which you'll hear a lot more about later, and the severity of depressive symptoms. So collectively, there is accumulating clinical evidence for a relationship between the brain and the immune system in depression. Biologically speaking, we're relying on an assumption that may not at first be immediately obvious, which is that the brain and the immune system are in balance with each other. It follows that perturbations to one arm could cause changes in the other. And we know this to be true from our everyday experiences. For example, anyone who's had a fever could tell you that being sick alters your behavior. We tend to withdraw socially, have decreased libido, eat less, sleep more, etc. It then wasn't a giant leap to link sickness behavior with depression, as behaviorally, the two conditions overlap quite a bit. Since the 1990s, groups have been studying depression, quote unquote, by manipulating the immune system to induce sickness behavior in various animal models. Similarly, the thought is that via chronic activation of the immune system, as seen in conditions like psoriasis and rheumatoid arthritis, these altered behaviors may become more permanent or fixed. However, not all patients who have an autoimmune disease are depressed and vice versa. So to explain this, we have to turn to another arm of our fulcrum, namely that there is this well-established relationship between chronic stress and depression. We tend to think of stress as being purely psychological, but of course there are physiological effects as well, including on the immune system. To illustrate, here we are once again at homeostasis. And perhaps you think to yourself, wow, it's a beautiful day. I think I'll go for a walk in the woods. You look around and seeing no one else within a safe 2-meter distance, let your hair down. But unbeknownst to you, somewhere lurking in the forest, there's a big, scary bear. Suddenly, you come upon this bear, and immediately your body goes into the fight or flight response. Evolutionarily speaking, it may be helpful for your body to ramp up an immune response so that any bites or injuries that you may incur during this encounter don't cause you to get infection and keel over before you can safely get away. So the acute stress of encountering a bear in the woods has suddenly mobilized your immune system in case you are attacked. And okay, that sounds pretty good, right? But there's sort of a problem. It turns out our so-called lizard brains aren't very smart when it comes to categorizing stressors. So the same system that's activated by a predator is also activated by everyday psychosocial stressors, like an exam or perhaps giving a talk. And we can understand this relationship through some relatively common physical conditions, including hives, shingles, or irritable bowel disorder. Physiologically speaking, we're really talking about the body's stress response, of which there are two main branches. One, the sympathetic nervous system, which targets lots of different organ systems through efferent nerves running along the spinal column. And two, the hypothalamic pituitary adrenal axis, which is outlined here. I think I can do this. Can you see my pointer?
Yeah.
Yeah. So hypothalamus, pituitary, and this is in the brain, and then leaving and going out to the adrenal gland. So both the HPA axis and the sympathetic nervous system stimulate the adrenal gland, causing it to release corticosteroids and norepinephrine, or adrenaline, respectively. So when your brain perceives a threat, like a predator or a psychosocial stressor, these two systems coordinate to prepare for fight or flight, but also any eventual wound healing and opportunistic infections that could come from injury. We know that this is true from decades of research showing that white blood cells of the immune system express alpha and beta adrenergic receptors, which bind adrenaline, and glucocorticoid receptors, which bind cortisol. For example, if you've ever used a steroid cream to deal with an itchy bug bite, it's the same principle. In some cases, large reservoirs of immune cells, for example, in bone marrow and lymph nodes, receive direct innervation from the sympathetic nervous system, so that when your fight or flight response becomes activated, they are perfectly situated to rush into the bloodstream and participate in whatever germ warfare might be necessary. For the rest of my talk, I'll mostly be focusing on this guy, which is a neutrophil, who we've already encountered before in the inflamed subgroup depression study. So you may be wondering, what is a neutrophil? If you were to look at them under a microscope, you would identify them by their characterizing odd lobular nuclei. They release large amounts of reactive oxygen species, and they also contain granules with enzymes that can remodel extracellular matrix, which I'll talk a little bit more about in a second. So the reactive oxygen species you can think of as kind of like a biological bleach where it kills pathogens. But they also respond to tissue damage. And so in that way, their role as extracellular matrix remodelers is important because they're basically helping to break down the scaffolding that supports tissues so that new immune cells can come in and build blood vessels to cause wound healing. Unfortunately, they're also well known for causing tissue bystander damage, as they're not particularly smart when identifying damage in a tissue versus pathogen invading. I put this here because I don't know if you've seen this cartoon series, Cells at Work, but it's a really fun way to learn more about the immune system. So I highly recommend checking it out. Okay, I mentioned certain tissues contain large reservoirs of immune cells, and this is especially true for neutrophils, which are the most abundant type of white blood cell in human blood. But I should also note that this is not true for mice. Neutrophils make up a large fraction of the bone marrow and respond quickly to stress signals. Here on the left, you can see the levels of neutrophils circulating in doctors who were either off critical care duty or on critical care duty. And as their perceived stress goes up, you can see that they have a lot more neutrophils circulating. This group also showed that that effect held true in mice. So now they've put their mice through a chronic stress paradigm. And you can see again that there are more neutrophils circulating in the blood. And finally, this is a really cool video. This is two-photon microscopy. So they're taking images in a live mouse. And what they're showing is that when you give an immune challenge, you see neutrophils leaving the bone marrow. And so neutrophils here are in green, and they're leaving bone marrow and entering the bloodstream. The authors then go on to show that this process is regulated, again, by the sympathetic nervous system. So we've talked about a brain to immune cell pathway, and our everyday experiences inform us about an immune to brain cell pathway. But clearly this is a complex system. And now, further complicating the issue, we have a new problem, which is that we don't find immune cells in the brain where they would ostensibly be positioned to exert an effect. The evidence for that can be seen here. P stands for parenchyma in this graph, and parenchyma is just a fancy name for saying functional tissue of an organ. So that's where you're going to have the neurons and the glia. And then they've looked at the parenchyma versus the brain's border regions, including the meninges and the choroid plexus, which I will talk more about in a minute. And ironically, this point was so sort of textbook taken for granted that no one bothered to do the experiment until a few years ago when this paper came out. But you can clearly see that there are, there aren't very many white blood cells present in the parenchyma, if at all, whereas you do find them in the border regions. And if you think about why this might be the case, it does make sense that we would have evolved this extra layer of protection for the brain, given that A, we were born with all the neurons we will have, and that these neurons make complex connections that are important for our survival. And finally, that for the most part, neurons can't divide to repopulate a tissue. So I want you to hold on to this idea that white blood cells can be found in the brain's barrier regions, because we'll be coming back to it. You've probably all heard before that the brain is an immune privileged organ. That actually has a very technical meaning and comes from some strange experiments where they put tissue grafts in a rat brain, but I won't get into the details of that experiment. If you're interested in further exploring the brain's immune privilege status, you can check out this review, which is really comprehensive and great. But usually what people are talking about when they invoke this concept of immune privilege is the blood-brain barrier, which prevents most things from leaving the bloodstream and gaining direct access to the brain. This includes both immune cells and the signaling molecules that they produce, which are called cytokines. So you can think of cytokines as sort of the neurotransmitters of the immune system. They help them communicate. And practically speaking, the barrier issue has been a very serious obstacle for pharmaceutical companies trying to develop new drugs for psychiatric conditions, because compounds that look promising in the lab fail to penetrate into the brain. So the real holy grail in this line of research is that we could manipulate something in the peripheral immune system, and that could then help people who are suffering from psychiatric disorders. And it's a lot more palatable to stick a needle in someone's arm than to drill holes in their skull. So to harness the potential of this system in treating psychiatric disorders, we really need to have a much clearer idea about what's going on at this brain-immune interface. In fact, there has been an explosion of interest in this tissue over the last 6 years, as you can see here. The meninges are implicated in everything from neurodegeneration to host defense, so neurodegeneration, host defense, to social behavior and anxiety. So what are they exactly? One of my favorite analogies came from my student at NIH who said that the brain is like a foot And the brain is the foot, the meninges are a sock, and the skull is a shoe. So in other words, the meninges help protect and cushion the brain and also have this supportive immune environment. Now, most people aren't very familiar with the meninges. It's kind of a niche area of anatomy. So I'll go through it in a bit more detail. The meninges are actually comprised of 3 separate layers. The dura, which is Latin for tough or hard, the arachnoid, which is somewhat self-explanatory and has to do with this web-like appearance of the arachnoid layer, and the pia, which is Latin for soft or tender. In humans, the dura alone is about 0.5 millimeter thick, whereas in mice, all 3 layers together are 20 microns. As the name implies, the dura are tough and fibrous, and they provide some structural support by sticking to the skull. It is important when reading the literature in this area to know that the dura are actually outside of the blood-brain barrier. So you can hopefully see up here in our dural blood vessel that there are these little holes. And so what they're indicating here is that these are fenestrated meaning window in Latin. And basically things can leak out of the blood vessels in the dura. The arachnoid forms this sort of sandwich space. So there are 2 tissue layers, a top and a bottom of the arachnoid. And within that space flows the cerebral spinal fluid. Whoops. Seen here. Finally, the pia is just a single cell layer thick, and it ensheaths the entire brain as well as the penetrating blood vessels. Together, the arachnoid and pia are called the lepto or thin meninges, and that is, again, as opposed to dura, which is thick. And the blood-brain barrier proper, as you might be familiar with it, exists at basically 2 levels. One is at the level of the tight junctions in the neurovascular endothelium, by which I just mean blood vessels in the brain, and also at the arachnoid dura barrier. So once a cell or a cytokine has made it into the CSF space, it's actually technically within the blood-brain barrier. The only thing that prevents these things from crossing over the CSF into the brain itself is a physical barrier called the glial limitans, which is comprised of these densely packed in astrocytes. Okay, so to sort out all of these complex interactions, we're going to need to rely on animal models because you can't go around biopsying people whenever you have some random pet hypothesis. So during my time at NIH, the model we used was chronic social defeat stress. In this paradigm, a dominant aggressive mouse is given time to set up camp and establish dominance in a cage, which you can see here, this is the white mouse. On day 0, a barrier dividing the cage in half is inserted, and our test mouse is put onto the other side of the cage. The next day and every subsequent day for 2 weeks, the barrier is lifted for 5 minutes at a time, and aggressive encounters are allowed to occur, and they are monitored by a trained observer. Otherwise, throughout the duration of the stress period, these 2 mice are co-housed together. So while the barrier prevents physical encounters outside of the 5-minute defeat window, it does allow for sensory cues like sight and olfaction. So there is a very psychological nature to this stressor, as it's kind of like being kept close to someone who bullies you all the time. At the end of the defeat paradigm, serum CORT levels are elevated and behavioral changes can be seen. I think it's important to point out that a major limitation to this model is that for all intents and purposes, it only really works in males. Female mice don't show this behavior. And if you put a female mouse on the other side of this barrier, instead of becoming angry, this aggressor will try to court her. But because of its popularity, efforts are ongoing to manipulate the aggressor to attack a female and thus induce the stress behavior. There are some species like California mice, which pair bond, and both males and females will show this aggressive behavior. but they lack the well-characterized genetic and immunological tools that we have available to us in the house mouse. Finally, I've put quotes around depression as a way of acknowledging that obviously we can't ask a mouse how it feels. So as with any animal model, there are always concerns about extrapolating our findings to humans, and rightfully so for psychiatric disorders in particular. Nevertheless, we have to start somewhere. So in the absence of talking mice, our readout will be behavioral assays. For example, anxiety behavior. This is the open field test. Basically, mice are balancing their desire to seek novel situations and a fear of predation, as mice are commonly preyed upon. And a stressed mouse, which is more anxious than a control mouse, will mostly hang out around the edges of the open field test, which you can see here. And this is a heat map of where they spend their time. So we quantify the amount of time they spend on the edges versus the center and how many times they cross. And you can see that the stressed mice show fewer crosses to the center. The other two behavioral tests I'll talk about today are related to depressive behavior. So if you're thinking like a mouse, what is it that you enjoy? As far as we can tell, one of the pleasurable aspects of a mouse's life is getting to know a new mouse. So we use this social interaction test to look at that component of their behavior. And basically, we plop the mouse into an arena that has one cup with a novel social stimulus and a similar cup with no stimulus. And then we just look to see how much time they spend investigating one versus the other. So you can see here in the control mice, the heat map shows they're spending most of their time around the social target, whereas the defeat mice are spending most of their time around the object. So we simply plot that as a ratio and call that the social interaction score. Similarly, for a male mouse, it turns out that they enjoy the scent of female urine. So we developed this urine scent marking test. You can see here, this dark purple spot that shows up is female urine that was pipetted onto paper. And we let the mouse run around this arena and they will mark with their scent. And then we photograph that and quantify how many markings they make around the female, where they are presumably advertising themselves to her, versus marking elsewhere. And you can see that the stressed mice don't really have any preference for the female, and further, they don't really mark at all. So both of these tests are looking at the anhedonia component of a depressive-like phenotype. Now that we have our behavioral assays sorted, next we'd like to know, is there evidence for an immune response in this model? And the short answer is yes. Previously published work from our lab at NIH and other labs have found evidence for this. Shown here, you can see that if you collect serum cytokines and assay them, there are a variety of them that are elevated, indicative of a pro-inflammatory state. When I joined the lab, I decided that I wanted to create a roadmap of the cellular response to stress by doing some rather simple characterization experiments. To do this, I collected a variety of tissues, including blood, bone marrow, skull, spleen, brain, lymph nodes, etc., from both defeated and healthy controls, and then I prepared the tissue for flow cytometry. Flow cytometry, if you're unfamiliar, is a useful way to characterize cells by exploiting the fact that immune cells express proteins that are unique to them on their surface. We can then tag those proteins with an antibody that's conjugated to a different fluorescent group. You've all probably heard about antibodies. They've been a lot in the news a lot because we're using antibodies to target the COVID spike protein. So hopefully that's a familiar concept. Modern flow cytometers can easily separate up 10 plus colors using different lasers. So you can look at a variety of cell types in a single go. You can also get information about cell complexity. So for example, on the y-axis here, we have side scatter. And this high side scatter is typical of neutrophils, which have that funky lobular nucleus that we saw earlier. Here I've shown a very simplified gating strategy to look at mouse neutrophils, shown in red based on the presence of their lineage marker, which is Ly6G. Now, I knew that eventually I wanted to visualize neutrophils using microscopy, but there aren't very good antibodies or stains for looking at them in fixed tissue. So we obtained this neutrophil reporter mouse called the Lysem GFP mouse, and basically they express GFP at a high level in neutrophils. And of course, whenever you're using a new strain or material, you'd like to test that it works properly. So we simply put these Lysem mice through our standard defeat paradigm and looked at the behavioral and blood immune profile. And we were happy to see that we recapitulated the behavior as expected, where they're both more anxious and exhibiting the anhedonic component of urine scent marking. And further, we found that they had increased numbers of neutrophils circulating in blood. So this was nice and agreed with the study I showed you earlier, where the stress mice had higher levels of neutrophils circulating with a different stress paradigm. We also noted that the blood neutrophil levels correlated with anxiety behavior, and there seemed to be a weak relationship with the urine scent marking test as well. So now we have an assay that we can begin to use to examine this arm of immune system to blood-brain barrier communication. And to explore this, we did meningeal whole mount preparations. So I've put up a picture of my trainee, Allison DePite, who is a future grad student. And this was truly a labor of love on her part because she took these images and hand counted each of these neutrophils for quite a few samples. And so what you're seeing here in gray on the left are the blood vessels, which are labeled with a dye that we injected just before killing the animals. And in blue are dots indicating where she's hand counted a neutrophil. The meningeal whole mount, excuse me, is approximately the size of your thumbnail from a mouse. To prepare this tissue, we deeply anesthetize the mice, then perfuse them with a buffer to desanguinate. Then we use paraformaldehyde to fix the tissues in place. And we do that with the tissue inside the skull. The skin and muscle are removed, the skull is fixed for 12 to 18 hours, and then we can cut away the top of the skull, gently peel off the meningeal layer, and mount that. So that has to be done under a dissecting scope. As I told you, it's about 20 microns thickness in a mouse. On the right, you can see what this looks like under high magnification. So DAPI is our nuclear stain. Tomato-lectin, as I mentioned, is what we're using to visualize the blood vessels. Lysem GFP is our reporter mouse for neutrophils, and then this is the merge. So what I hope you can see is that in the stressed mouse, there are a lot more of these neutrophils than there are in the control. And here you can see the quantification of what I just showed you. So as we expected, there was a stress-induced increase in the meningeal neutrophils seen with immunohistochemistry. To verify our IHC results, we once again turned to flow cytometry. And now we are just using wild-type mice as they were more readily available. We used a panel that could broadly identify lots of different types of immune cells, but as you can see, just like in the Lysem mice, there was an increased number of blood neutrophils in wild-type mice following stress. And further, there was an increased number of neutrophils in the Meninges. We did look at the relationship between the two tissues, and there wasn't anything significant here. If anything, maybe as blood neutrophil numbers go down, they're increasing in Meninges, which could suggest there's some infiltration, but we don't know for sure. We also wanted to know about the meningeal distribution in an acute versus a chronic stress paradigm. So to look at this, we simply cut short the number of days the animals were exposed to the stress. So you can see animals either got 1 encounter, 2, this is and another encounter on day 4, 8, and 14. So at each time point, we perform the defeat and then sacrifice the animals 2 hours later to look by flow cytometry at their tissues. Perhaps this is a bit comforting, but an acute stressor was not sufficient to drive accumulation of neutrophils in meninges. It really took that chronic nature of stress before we started to see a change. Whereas in the blood, it happened relatively quickly. Now, to our knowledge, there's nothing in the literature about the role these meningeal neutrophils play, either in healthy controls or a stress situation. So we next set out to characterize them functionally. And to do that, we used single-cell RNA sequencing. So this is a very sophisticated way to take cells from any given tissue and examine them individually. And the benefit is that you're able to look at all of the mRNA transcripts in a particular cell and know which cell it came from. So we did the experiment, and here you can see the results. Ultimately, we ended up with 8 pooled individual home cage animals and 4 pooled defeat mice. So the data here is, here on the left, is representing all of those mice together. And first we set about identifying what the cell clusters were. So we were able to assign identities to these clusters and noted that we had a diverse array of different types of white blood cells. And this is a t-SNE plot, which if you're not familiar with, I'm happy to talk more about, but basically it allows you to take very complex data and collapse it down to look at more simplistically in a graph. And the distance between clusters doesn't necessarily have that much meaning. It's a way of grouping things that are more similar to each other. So in any event, you can see that we got this diverse population of cells. And when you assign the group back to these different cells, so either home cage or defeat, you see once again that there is a relatively increased proportion of neutrophils in the stressed mice, shown in lilac here. We also noted that the neutrophil cluster could be subdivided into different clusters, and this seemed to map onto neutrophil maturation. So the meninges is located very close to the skull. We had some concern that there could be skull bone marrow cells leaching out. And if those are present in our sample, they're likely to be these pre-neutrophils up here. But what you can see is that we get an array of different phenotypes that represent less mature up to more mature. And we know that based on the gene expression that you can see here. And it's not necessary to look in at the individual genes, but you just get a sense of the changes in gene expression. If you look at the relative abundance of each of these subtypes of neutrophils and compare home cage to defeat, we plot the ratio here. And you can see that it's really the mature or the more mature forms of neutrophils that are most impacted by stress. We also I have this graph here showing that ribosomal or translational machinery is suppressed, which is typical of neutrophils that are differentiating from an immature state into a mature state. Next, we had this wealth of information available to us about gene expression differences. So here on the left, I'm showing a volcano plot. This allows us to visualize all of the differentially expressed genes between home cage and defeat animals. And now we're looking at the entire neutrophil cluster. So not subdivided, the whole sort of cloud of neutrophils. As you get further out on the x-axis here to the right, these are genes that are more highly expressed in defeat animals, and these are genes that are more highly expressed in home cage animals. But it's hard to discern what this means, just kind of eyeballing them all at once. So what we do is we take our list of differentially expressed genes and plug that into software that tries to predict what biological pathways might be associated with the changes you're seeing in your data set. And so that's what's represented here on the right in the neutrophil reactome. And what we noticed was that there were quite a few pathways related to actin filament processes and cellular size. So actin is kind of the bones or the skeleton of a cell. It gives it structural support. We wanted to have some confirmation of what we were seeing in our gene expression biologically. So we turned to this image stream analysis platform. So image stream is a way of doing a combined flow cytometry and microscopy experiment. So what you can see here is that the cytometer is able to take a picture of the cell as it's moving through a stream. And then you can stain the cell as you would for a normal flow experiment with a variety of markers that help you define the lineage. And we added in phalloidin here because that stains actin filaments. So unfortunately, we didn't see any discernible changes in actin polymerization, which the bioinformatics predicted we might, but we did see an increase in the percentage of enlarged neutrophils in CSD meninges, shown here. But we weren't really sure what this could mean. However, there was a paper that came out fairly recently that was also looking at neutrophils, and they were using a mouse model of Alzheimer's disorder. And what they noticed was that in the fine capillary bed of the brain, they were seeing these neutrophils get stuck and stall, and that was actually sufficient to block the flow of their blood flow tracer. So you see in red that there's a gap here right behind this green neutrophil. So they quantified the number of events and found that there was a difference between wild-type mice and Alzheimer's mice. They also did MRI to look at cerebral blood flow. So you can see in a wild-type mouse, you have blood flow, or shown here by a lighter color, in the cortex. But in the Alzheimer's model, there is reduced cerebral blood flow. However, when you deplete neutrophils with this Ly6G antibody, you get a recovery of blood flow. And that's quantified here. So this was really exciting for us. We thought maybe we could apply what they learned in this paper to our model, where we're seeing an increased size in neutrophils. We went back and looked at the brains of the animals that I showed you previously. So these are the animals that we did IHC on Meninges for. And we found that no matter where we looked in the brain, there was an increased number of neutrophils. But it's important for me to point out that we never saw neutrophils crossing over the blood and entering the brain parenchyma. So all of these neutrophils that you're seeing represented in blue dots are stuck in a blood vessel. And you can see that here in the merge. And here's the quantification showing you again that this did not seem to have any regional specificity. Next, we took a stab at doing a two-photon experiment. This definitely needs to be replicated. It's very preliminary, but I thought that the videos were really neat, so I wanted to show them today. Basically, what we did was we collaborated with Dorian McGavran's lab at NINDS, which is the Neurological Disorder and Stroke section at NIH. And they are able to do these thin skull window preparations so that there's no damage to the brain or the meninges. And once again, we're using the Lysem-GFP neutrophil reporter mouse. And in this case, we've injected Evans blue, which is a dye that helps us visualize blood flow. So in the control animal, what I hope you can see is that there are these quick flashes of green as neutrophils go through the fine capillary vessels in the brain. And if I play that again, yeah, so hopefully you can see that. Whereas in the stressed animal, what I want you to focus on is right here. So we were seeing these events where neutrophils were not only not zipping around like they were in the home cage animal, but in this case, they're actually sort of meandering back and forward along the blood vessel. And further, there was this sort of hazy effect where perhaps the dye was leaking out. So we were really excited by this finding, and it's something that needs to be replicated, but is an interesting sort of follow-up to this idea that there may be a connection between neutrophils and blood-brain barrier damage. Show you that one more time. Okay. So to look at the functionality, right? So we've seen that there are neutrophils present, but we still don't really know what they're doing and how that relates to behavior. And what we really needed was to deplete them in some way and then assay behavior to see what the effect was. This was a 2.5-year effort where we ultimately discovered that the stressed mice didn't do well if you totally deplete neutrophils like they did in the Alzheimer's mouse. So what we settled on was manipulating receptor-mediated tissue entry. And so what I mean by that is imagine that you're a neutrophil and you're floating through the bloodstream, and all of a sudden you encounter a receptor on an endothelial cell lining the blood vessel that will allow for the neutrophil to traverse into the tissue itself. And so what we hoped was that we could pre-treat the neutrophils with a blocking antibody. And then when the neutrophil encountered that receptor, it would no longer be able to cross into the tissue. And so we injected this blocking antibody before starting the defeat paradigm, and then every 2 days thereafter. So the blocking antibody is on board throughout the duration of the stressor. And I also want to say, for those of you who are getting into science, persevere. It can be worth it. This was my experience. So the hypothesis is our antibody drug should block tissue entry. And indeed, that was what we found. So here you can see that in the vehicle controls, we have our now many times replicated increase in neutrophils. But in the animals that got the antibody, that increase is blocked. This was true in the Meninges, but it was not true in the blood. And sort of the coup de grace, what we were really excited about was that when we looked at behavior in these animals, there was a partial recovery of social behavior in the animals that got the blocking antibody, whereas those that got vehicle were just as asocial as we would expect them to be. It's interesting that there's this sort of binomial distribution. We don't yet know what that means, but we're exploring that further. And it's also interesting to note that these two behavioral components, anhedonia and anxiety, are not necessarily showing the same effect. So you can see that there's no change in the anxiety behavior of the animals that got the depleting antibody. To summarize all of this, we have learned a lot from our mouse model that indicates perhaps neutrophils are playing a role in the progression of depressive symptoms. And it's interesting that sort of independently of the work that we were doing at NIH, Ed's lab and the consortium that he heads up had also found that neutrophils correlate with depression severity. And so the hope, of course, now that I've come to Cambridge, is that we can take the lessons learned in a mouse model and apply them to the human situation, which is, of course, a lofty goal, but you have to have dreams, I suppose. So future directions, I'm hoping to follow up on these promising leads. Now that I'm here working with Professor Ed Bullmore and the NEMA Consortium, which is the neuroimmunology of mood disorders and Alzheimer's disease. I've put links to the website for both the NEMA Consortium and an ongoing clinical trial looking at inflamed depression in case you're interested. And what I hope to look at is whether there's any conservation of the pathways that we found in the mouse model between, and whether we could some way manipulate those to help people suffering from depression. And so with that, I will thank both my lab at NIH and my former supervisor, Miles Herkenham. Especially thank Ali DePite, who did a lot of work with hand counting images. I'd like to thank my supervisor here, Dr. Ed Bullmore, as well as collaborators and core facilities that facilitate all of this work, and our funding, which comes from the Biomedical Research Center. And also, just if you're curious, all of the uncredited images were made in BioRender, which is a free program, and it's really easy to use. So check it out if you have a talk. And with that, I'll also thank the Trinity Postdoc Society and Science Society for inviting me, and take any questions if you have them.
Yeah. Dr. Kigar, many thanks for your talk. And if there are any questions, either write them to the YouTube chat or use the raise hand button in Zoom. And yeah, there is one question.
Hi, Stacey.
Hi.
Thank you for your talk. It was so great. And I really liked your slides. They're really pretty. Yes, so I have one main question. You showed somewhere in your slides that acute stress is not sufficient to drive neutrophils into the meninges.
Mm-hmm.
And then the other evidence was that neutrophils don't actually migrate into the parenchyma.
Right.
Right. So are you suggesting, or rather is the evidence suggesting that neuroinflammation is not a thing in inflammation-linked depression, and i.e. inflammation is only isolated to the periphery?
We're only looking at cells here. So there's the caveat that the cell, the neutrophils in blood go up immediately. There could be products that they make that we are not detecting by flow cytometry that could be causing neuroinflammation much earlier. We wouldn't capture that in this type of assay. So that's one thing. But I do think it's interesting that this particular class of immune cells, neutrophils, which are kind of famously known for causing bystander damage, don't, apparently an acute stressor is not sufficient to drive them to accumulate in this area. And maybe that's a good thing. But again, it doesn't necessarily mean that there aren't other changes. And I've only talked about neutrophils today, but you can see in this graph that other cell types are also changing. So you can see, for example, that B cells are dipping down, and that's happening pretty early on. So I'm sorry, I forgot to explain that on the y-axis here, these are days of defeat. So 0, 2, 4, 6, 8, 10, etc. Monocytes are another immune cell, and they are increasing pretty rapidly, right? So I don't mean to myopically just focus on neutrophils and say there is no neuroinflammation. There probably is. Does that answer your question?
Yeah, absolutely. Thank you.
Hi, Stacey. It's Ed. Thanks very much for the talk. Beautifully clear. So I had a sort of similar kind of question, which is, particularly when you think about the difference in response to acute and chronic stress, and it's about the life cycle of neutrophils. They're quite short-lived cells. I just looked it up on Google. It said their life cycle is about 5 to 135 hours. So 4 or 5 days max. And I'm wondering, given that there does seem to be a kind of effective chronicity, where do you think the memory of stress exposure sits in the immune system? Because presumably it's not in the neutrophils because they're too short-lived.
That's a great question. We've thought a lot about that. Can you see this? So this is an experiment. Again, it's kind of a one-off. But what we did was we did whole skull clearing. And you can see that, well, first of all, just to orient you, on the top, we have bone marrow and the skull. And then there are these blood vessels that directly connect the bone marrow in the skull to the meninges. So what I think is pretty evident is that in the stressed animals, there are a lot more neutrophils in the skull bone marrow. And I suspect that chronic stress could be encoded in stem cells, which would have more of a memory capacity. And so something, I mean, this is speculative, but something could be changing in the hematopoietic stem cells so that they're more able to proliferate or have a different transcriptional signature that allows them to be recruited, et cetera. So I think that would be a really interesting thing to follow up on. Does that answer your question?
Yeah, it does. Thank you.